Hello and welcome to the Virginia IGM. In this series of lectures, I'm just kind of going to go through what's already been kind of published out there and just tell you kind of what I think about it as we're kind of going through it. So bear with me on this one here. For this particular workshop plan that we're asking you to look at is we're looking at genes, backbones, and basically the biobrick structure. Normally these would have a lab associated with it, some DNA extraction. We'll get a chance to do that a little bit later at Skyline College. But basically what this is going on to tell you here is, if you look over here, this is chromosome number 1, which is made up of 246 million base pairs. Very big, lots of stuff going on there. And as you remember from our talk before, that within our cells, this is a eukaryotic cell, we're going to have a nucleus, and in that nucleus you're going to have the genes, which are located on chromosomes that look something like this, and those chromosomes are arrangement of DNA and protein that are held together, and we have this genetic sequence. Now that's basically a gene. And so we would say things like, people say things like, you have the gene for blue eyes. No, you have a gene for hair color. So there's many genes that are involved in controlling the color of your eyes. But you have an allele, an alternate form of that gene that causes that. And so what happens is these genes produce a protein. And so when you have blue eyes, the proteins that you have enzymatically create a structure that is blue. And so ideally what we're going to be doing with the IGen system is kind of moving genes around and creating things that we want to do. Now the idea is to kind of use these genes kind of like a circuit, a little bit like electronics. I don't know how much you know about electronics. I don't know how much you know about biology. But what's really kind of interesting is you can do things like make a genetic toggle switch, an on-off kind of thing, and you can make a genetic, what we call a repressilator, an oscillator. And so what's going on here is that we have these genes over here, and what happens, this LAC-I gene represses this gene represses this gene here. So in the presence of heat, we will actually turn this gene on. In the presence of this IPG, we will turn this thing on. So this basically what this does is, depending on what we want it to do, whether we put heat here or whether we add IPTG, we can express these kind of different proteins. So this green fluorescent protein, we could turn it on or turn it off. And so in the presence of IPTG, it would be on. However, even in the presence of IPTG, if we put heat on this, it would shut it off. And so these here are what we call, on the parts of the gene, these are called promoters, and these are called repressors. And so they make the proteins. And we'll talk about that a little bit more. But here's a great thing. If you want to do something genetically, we could turn it on and we could turn it off, and we could have it kind of flip around. Now the oscillator over here, what we're looking at, does something a little bit different. This gene over here stops this gene over here, stops this gene over here, and then this gene over here stops this gene over here. And so what's going to happen is A represses B, B represses C, C represses A. And so what happens is you can have sequentially go on. So A would be on, and then B would be on. The A would shut. A would be on, and then C would be on, and then B would be on. I'm losing it. But anyways, as we can see, we can switch back and forth between these three different genes. And so it's something very interesting that we can do and see. And again, the reason is because it's a little bit different than electronics, is that it takes a while for these genes to be transcribed and translated. And so while this is working for a little bit, then we'll shut this one off, then we'll shut that one off, etc. And we'll go on it a little bit more. So the gene construct system typically that we see is that we would have a promoter on the sequence. This promoter would bind to a ribosome binding, this promoter would make a piece of RNA, and we talked about that messenger RNA. This messenger RNA is going to have a ribosomal binding site on it so that it will bind to the ribosome, and then the ribosome will actually make this gene. And remember, we have to have an ATG start site, and then we have a terminator here. what this is looking at here, this is another construct. How do we know if this gene that we're looking at produces a product that we can't see or that we can't measure? Very interestingly, what we can do is we can know that the gene is there and that it is being transcribed and probably translated, is that if we have this setup. So we have a promoter on the DNA that will make sure that a messenger RNA is being made here. That messenger RNA then is going to have the ribosomal binding site for the gene of interest, and now also it's going to have the ribosomal binding site for green fluorescent protein. And so if we see the presence of green fluorescent protein, then pretty much we're going to assume that our gene product is going to be there. And so that, if it's not working, then we know a little bit that it's not due to something else. And so what can you do with things like this? And so here are all the different colors of fluorescence. So the green fluorescent protein, which is basically this one right here, this green fluorescent protein, what they did is they genetically engineered it so that it would have colors from basically this cool blue to this kind of plum color, and an interesting student can actually grow these up and make a little here's a little sunset in here. Again, you can put those in fish and have fish of a different color. Here we have all these different colored fish inside of here, so that's kind of a fun thing to do. It's kind of fun. So let's do it for science and see what else we can do with it. Well, basically, so when we have this palette of colors, what we'd be able to do with this palette of colors, and in here we're looking at the phases of cell division and mitosis, and we can have one thing stained for DNA, one thing stained for the cytochrome, and so where these dyes are where different things are happening. And so we see the chromosomes here, we see the microtubules on the other side, and we can have a good idea of kind of what's going on in the cell. So this is a tool that we can make to do many things inside the cell. Now, how we're going to use this tool genetically is by hooking up the DNA that we have of interest to a plasmid. And so here's an example of an electron micrograph showing the plasmids and your host genome. And so it's a problem, and Chris said this in class, is that the host genome a lot of times doesn't like having extra DNA inside of it. It tends to spit it out. And so what we found in nature, that there's these things called plasmids that are separate from the bacterial DNA, from the host chromosome, that will get replicated, that is copied, and they will go from generation to generation. And what we can do is we can put a gene of interest on these plasmids. The other great thing about these plasmids is it's very easy to purify them and very easy to kind of share them. And so when we get our kits, we will be obtaining a bunch of these plasmids. Each little vessel will contain over 360 different plasmids. So I was going to look. I don't have one of those kits available right here. And so what happens is what the BioBrick has done is they set up standardized what we call a plasmid backbone. And so how they give you a library of these genes, and we will get a library of almost 1,000 different genes to do stuff with. I'm really excited about all those colored pictures there. But so here we have a kind of schematic or a map of a plasmid. This needs an origin of replication so that it will cause itself to be replicated or another copy made. It has an antibiotic resistance gene that we talked a little bit about in this class. That tells us that we know that the plasmid is there. And then we'll have a gene, and the gene is cloned into a specific site. And these are restriction enzyme sites here. And what this allows us to do is to kind of put a gene in, and allows us to take a gene out. Now, these genes that we have cannot have these restriction enzyme sites inside of them. And so this is a really good tool for moving this about. And again, if our gene has it in there, we could always, there's different ways that we can do to move things out. So if you're not familiar with restriction enzymes, they're kind of like the scissors of our tools to manipulate DNA. And so what's going on here is here's a circular plasmid representing a circular plasmid. Here is the restriction enzyme EcoR1, and what it does is it introduces a cut into the DNA, that double-stranded DNA, and it leaves kind of a sticky end. So here's the EcoR1, it cuts over here, and what it does is 5' to 3', and this is 5' to 3' going the opposite direction. It leaves this AATT single-stranded end, and this TTAA single-stranded end on the other side. They're going in opposite directions, and so they're complementary to one another, and so they will bind to one another. Uh, Again, Chris called it magic. It's not magic. It's just basically hydrogen bonds. They will come in. They form a shape, and they will bind to one another, and they'll hold themselves there for a little bit. So you can have these two pieces of DNA in a solution, and they will come together right at that site. Really, truly amazing. And the thing is that we have a number of these tools. We have EchoR1, which cuts in this sequence. We have EXPA1 that cuts in this sequence. We have SPE1 that cuts in this one, and PST1 that cuts in here. And if you notice, what's really kind of interesting is EXPA1 and SPE1 actually have overlapping fragments that are similar. So they'll come together, and they will form what's called a scar. I'll talk about that a little bit later. But it's a way to kind of clone something in there, and then EXPA and SPE1. So we will be using a lot of those restriction enzymes. Case study, FITZ and mini-cell formation. I don't remember that. DNA extraction. And so I'm not going to go over those case studies. I'm not going to go over the DNA extraction in there. We will be doing some of that lab work, but again, so this is the complement. So you learned about transcription, translation, what's going on with those, and then we learned a little bit about how we have a DNA moving our gene-like product. Okay? So have a good day. Hey, do you like my office? Pretty fancy. All right, catch you later.